Hello, I'm Professor Tony Mok from the Chinese University of Hong Kong. Thank you for the opportunity to ask me to talk about this important question: is the resistant third generation TKI fueling a new therapeutic strategies? Now, so first of all, this is my disclosure, and more specifically, I'm on the board of director of these three companies. Any question you ask me, just like, will you marry me? Or any other question, is yes, no, or maybe. However, to answer this question is very simple. Definitely, it's not a no. It's either yes or maybe. So I'm going to go through some of the biology and the strategy to answer this question. First of all, we have to understand the biology of the strategy, and then subsequently focusing on the targeted therapy strategy, the ADC strategy, and the immunotherapy strategy after failing osimertinib or other third generation drug. First, about the biology. Now, the biology of resistance is complex. I'm trying to summarize the essence of it. Now, despite of what drug you give, when EGFR first presents itself, it's not just a single EGFR mutation. Now, this is a study by Frampton Cancer Discovery from Foundation Medicine, over 4,000 cases. The patient with the EGFR mutation, as you can see, also had a number of co-mutation in the beginning. That may include TP53, which is the most common one. But other than that, you can have a whole arena of different mutations, including ALDA1A and also the CTK. So how much impact of this co-mutation is not certain, but then there are some prognostication factor of each of this mutation. And also, we know that once you put the drug EGFR mutation prostate cancer in stress, there will be having different kind of response in terms of the subsequent mutation. Now, this is the data from Singapore indicating about close to 20 cases of patient with EGFR mutation. As you can see, the trunk mutation is consistent, but then the branch mutation of this EGFR can be quite diverse. So in other words, it will be hard to predict what may happen once you put the stress on the EGFR trunk mutation. So we'll predict it to be a very heterogeneous in terms of outcome as well. So for that, is actually come into some of the data that we have. The data has come from the plasma-based test by Guardant 360 on the patient who have first line or second line osimertinib, namely the plasma sample from the FORA study and the plasma sample from the AURA study. So as you can see, consistent theme is that you have the C797S, and also you may have a HER2 amplification and a MEK amplification. The rest is a little bit heterogeneous, and then a large proportion unknown. Why unknown? Well, we have to understand that we are actually using Guardant 360, which only have about 71 gene. So in other words, there can be a lot of so-called mechanism that's not covered. But one other important point is that patient who are second line osimertinib, meaning they are start off with T790M positive, about 50% of them will lose the T790M as one major mechanism of resistance. So putting that in mind, I think one conclusion is that we get multiple resistance mechanisms. Therefore, we need multiple strategy. It's unlikely that we have one strategy to cover everything. So on the multiple strategy, let's take a look at the whole picture. Now, this is a simplified picture from Cancer Discovery. And you can see that it can be a resistance mechanism in the EGFR, which is the C797S, or it can be in the MEK pathway, which we already talked about briefly. And then HER2 has also covered it. But then there's also the EMT transformation, meaning that transformation to small cell lung cancer. So those are the mechanisms that we'll talk about today. And then also that we can potentially target a apoptotic pathway, but right now we don't have a very good apoptotic drug, like the BEAM2 is actually important on the apoptotic pathway. But overall, we also have to cover the ADC and the immunotherapy. So that is the basic scheme of our discussion. Now, targeted therapy, what is the strategy? Well, the first is a strategy against the C797S. Now, we have to understand that the C797S is the binding site of osimertinib. So once it got so-called mutated from C to S, then it becomes a resistant or not non-binding to osimertinib. But in order to tackle that, we have to be very careful because some of this patient also have the T790M mutation in the same time, which prevent you from binding a T790M site. So we end up that we require something called the aerostatic compound. So this is actually the first paper come from Dana-Farber that indication of a allostatic compound potentially that can inhibit a patient with C7IFNS. Moving forward, there is one of the first data that has come out subsequently on an allostatic compound. It's called a compound 40125 by JBJ. And then you can see that it is an anchor on one side and then inhibiting back to the pocket. And however, this has to be in combination with osimertinib for the proper inhibition of the tumor. So this is actually data that was from 2019, but up to this day, we have not any solid data on its clinical efficacy yet. On the other hand, Blue 947 is a compound as such targeting the T790M and C797S. And for that, you can see this compound can actually on the cell line and it had a reasonable IC50. And right now, this drug is actually ongoing as a clinical trial. Now, the other strategy is to inhibit the EGFR and MEK in the same time, and that is the bi-specific antibody, amivantamab. You probably know a lot about it already. And so the early data is relatively encouraging from the Chrysalis study, phase 1. And on the patient who failed that osimertinib is a combination of amivantamab together with lasatinib, a third-generation TKI from Korea. And we're looking at tumor response rate. It turned out to be 36%. Reasonable response rate in a small population, but the progression-free survival about 4.9 months. But most of these patients had multiple previous line of therapy. So early data, not quite ready for so-called the major treatment yet, but then it is one strategy that should be developed further. Now, the next question is that whether we can really utilize the biomarker. So they had a very small subgroup of 10 patients with IXC overexpression of either EGFR or the MEK, and for that, out of that, 9 out of 10 patients had responded. But again, small population, whether that will be the future biomarker, certainly to be validated. But one very important strategy on the MEK inhibition. Now, for that is another way inhibited is by tyrosine kinase, and for that we already got cometinib and tepotinib, which is a potent type 2 MEK inhibitor that is actually approved for MEK exon skipping. But what is its application in a MEK amplification post-EGFR TKI failure, such as osimertinib? So the data actually come from the Tatton study from this year's WCLC. Han reported that fact that the patient who failed the osimertinib and then with MEK amplification defined it as either FISH copy number over 5 or the MEK CYP7 over 2, IXC 2 plus or 3 plus or NGS positive. And the 4 cohort is either prior EGFR failure with the osimertinib or first and second generation TKI failure but T7M negative, or first, second generation failure but then T7M positive. Cohort D is same as part B2 except that they give a low dose of sunitinib, which is related to toxicity. So for that, the response rate is actually for osimertinib failure group is actually impressive, about 33%. And that is a patient who failed osimertinib go on to receive a savolitinib, which is a MEK inhibitor together with osimertinib. They still got 1/3 of the patient responding. But on the other hand, if the patient are actually first and second failure, but then T7M negative, they're still able to induce a response of about 60%. And that is actually translated to a certain degree of improvement in the progression-free survival. But this needs to be validated in a randomized phase 2 study, which actually will roll out sometime this year. Now, based on that, let's go on the next strategy, which is small cell transformation. The data on the small cell transformation is scanty, but this is one of the best papers we can get from 2019. 58 patients with small cell transformation. At the time to transformation, about 18 months, but most of them, almost all of them have TP53 and RB1 loss. But the response rate is initially quite reasonable, 54%, but the median progression-free survival is short, only 3.4 months, and overall survival is only 10.9 months. So relatively disappointing data. So once you get transformed, the prognosis is poor. So the question is that the strategy would be, can we really prevent it from transforming? So for that, there's one ongoing clinical trial right now in Memorial Sloan Kettering by Helena Yu. Is a patient who are EGFR mutation positive, but in the same time, RB1 loss and P53 positive. And then the patient may have the TKI started or the treatment naive within 6 weeks. Then they were able to get combination of osimertinib to get a standard chemotherapy or etoposide and platinum. So that's the question, is to give the chemotherapy early so where they can prevent the transformation if the patient is RB1 lost and P53. So that's one strategy for small cell transformation. Now, after this targeting the EGFR, MEK, and also the small cell transformation, let's talk about ADC in general. The concept is important. In a way, it's a monoclonal antibody targeting the cancer cell with a specific antigen against. And then you use a linker to link on a cytotoxic drug, a very potent one. Usually is the Toucan is one of the topoisomerase inhibitor. And then this linker is actually, released it, or actually resolved it once in endosome, and then released the cytotoxic drug to kill cell. It sounds like perfect system, but of course, nothing is perfect. But let's take a look at the potential efficacy. The major development now of ADC is the fact that one molecule can have multiple cytotoxic drug, meaning that the linker ratio is actually higher, meaning that they can carry more cytotoxic drug with it. So this is the list. If you're focusing on the lung cancer in the HER2, we have the TDM1, which is a long time ago, and then the new one, trastuzumab deruxtecan, while ADS-8201 is actually recently approved for breast cancer as well. And then there's a HER3, the U3-1402, and there is a CMAC one, and then also there is a TROP2 one. So basically, the three site that ADC may target is the HER2, HER3, and MEK, and actually TROP2. Now, let's look at the data. With the U3-1402, which is anti-HER3, now almost all EGFR mutation positive cancer patients will have an overexpression of HER3. So that makes sense for it to be a recent target. And then the data was actually reported in Cancer Discovery by Pasiani, first released it in ASCO, subsequent in Cancer Discovery, reporting a response rate of about 39%, and then median progression-free survival 8.2 months. So it is early data, but still, that is a very suggestive data. And so this drug is actually moving on into randomized study right now. So the likelihood is the fact that whether it's going to be treatment naive post-osimertinib or post-osimertinib and then post-chemo, and then so this type, either of this randomized study likely will roll out sometime this year as well. So this will help us to define what is the best timing to consider the ADC in the osimertinib failure. Now, this one is on the HER2, but then mostly it is on HER2 mutation. Now, breast cancer is HER2 amplification. In lung cancer, post-osimertinib, we may also have HER2 amplification. But the data we see a 55% response rate with the trastuzumab deruxtecan is on the HER2 mutation, and then with the progression-free survival about 8.2 months. Those are attractive, but its approval for HER2 is on the way, but its application in HER2 expression is still pending to be defined. There is a small subgroup study from this one, and response rate is about 24% reported by Apikawa the year before. Now, so with this in mind, I think that we have to wait for more data before we can decide. What about the so-called ADC on MEK? Now, ADC on MEK, this is data come from Ross Cambridge from WLC this year. Trastuzumab vedotin, and then they look into the MEK amplified patient, but then I'm not quite sure the definition from this abstract. And you can see that they got a group of a patient that is EGFR wild type and a group of patients that EGFR mutant, 60 in each group. And there's high and intermediate in either group. Now, to cut the story short, for patients without the mutation that are MEK high, the response rate is actually reasonable at about the 35%. But then for the EGFR mutant, the response rate for the MEK high is only 13%. But to remind you, the patients who are mutant, they actually have multiple line of therapy, and then they have chemotherapy just beforehand. 84% of them had chemotherapy. So I think this is the early data. I will not give up entirely yet, but the early data of 13% response rate is not very supportive of this direction for EGFR mutant. Now, another was TROP2. TROP2 is in respect of the EGFR mutation status. It may occur in a lot of different types of cancer, including lung cancer. And then the so-called the TROP2 expression is independent of the EGFR status. And this is a glycoprotein that's common across all cancer. Now, for triple negative breast cancer, Trodelvy had already been demonstrated response rate 31% and are already approved for breast cancer. And median progression-free survival is about 5.6 months. So exciting data. But for lung cancer, it is still early, and there's still only phase 1 data that's being available. On EGFR mutation positive, I think we still have to wait and see. Now, last but not the least is immunotherapy. One thing we must be aware that TME status, the tumor microenvironment status of the EGFR mutation, is different from other cancer. The tumor microenvironment is created by a tumor for the purpose actually to protect the cancer, because the cancer itself want an environment that they're comfortable to grow in. So the player may include the lymphocyte, the myelocyte, the cytokine, the fibroblasts, and also the multiple of the exosome. And all this environment is that have the purpose of to promote cancer growth and also to defend against the immune cell. So, and especially in EGFR mutation positive lung cancer, it's quite well known that the environment is actually quite resistant to the PD-L1. So, how may it affect it? This is a good summary. There's a few aspects of the EGFR that is actually special. So first of all, is the fact that there is a CD73 and adenosine, which is usually upregulated in EGFR mutation positive. And adenosine is the one substance that may potentially inhibit the myeloid cell from activity. The T regulatory cell is also an important part of the whole picture, and the EGFR mutation is actually a reduction of the T cell degradation. And also EGFR may potentially affect the PD-L1 expression, usually reduce the PD-L1 expression. So all this putting together, making EGFR mutation probably a poor response to immunotherapy. That is a hypothesis based on science, based on biology. But then when you look into the clinical data, this is a report from Julian Mazzazier on the multiple different type of patient with the driver oncogene, and EGFR mutation, the response rate in the second-line situation is only about 12%. And that is actually related to a progressive data in the subgroup of the randomized study, like the Checkmate 017, 057, and the OAK study, and also the POPPER study. All this includes a small percentage of the EGFR mutation population. And you can see the consistent response rate about 10 to 15%, and the median progression-free survival about 3 to 4 months. So overall, it's quite well established as a single agent, either retrospective study or prospective study, that we do not expect a good outcome. So once you talk about the microenvironment, there are multiple factors in play. And one that we can actually contribute is actually angiogenesis. As you can see, the VEGFR contribute to a number of so-called mechanisms in terms of the microenvironment. So potentially target VEGFR may facilitate and change the tumor microenvironment for the immunotherapy. So this is first observed in IMPACT 150, as you probably know, is Taxol, Carbo, bevacizumab, and tislelizumab. And then in this particular study, 10% of patients are EGFR mutation positive. So for that subgroup, there is a high response rate, about 71%. And when you compare the 4 drugs with the Taxol, Carbo, and bevacizumab, there's improvement in progression-free survival and overall survival. As compared to 3 drugs, only the Taxol, Carbo together with Atezo compared with Taxol, Carbo with the bevacizumab, there's no such advantage. So look like there is a so-called improvement when you give bevacizumab together with a PD-L1 inhibitor. Now, we need further data. And so there's one additional study this year, last year, is the Orient 31. That's come from China, EGFR mutation positive patient who failed the TKI. Looking at the patient that are actually received 4 drugs, which means that patient actually received the cetuximab, which is a anti-PD-L1, and also the a biosimilar bevacizumab, and compared to the Taxol, Carbo alone. And for that, you can see that there's improvement progression-free survival, 6.9 versus 4.3, and improvement in response rate. So, and additional study from China showing the same phenomenon with the Forte regimen. It will be first substantiated by IMPA 151, which is also being done in China. 50% of this population will be EGFR mutation positive. Another one is to target the adenosine inhibition. You can either target the receptor or the CD73 by a drug called anucumab. Now, based on a study called co-study that come out just this year on stage 3 disease, durvalumab alone as a maintainer Pacific versus durvalumab plus oleclumab versus another immune inhibitor, we're able to see the higher response rate as well as an improvement progression-free survival. Now, these are not EGFR mutation positive patients, but just demonstrate that potential combination of a PD-L1 and an anti-CD73 may improve the treatment outcome. So I think it will be interesting to look for those data in EGFR mutation population. So coming back to the question, well, yes or maybe, that's definitely, it is a yes. So I think that we should move on, and then to summarize the fact that we have the biology to demonstrate why osimertinib had failure, and then we targeted including strategy on C797S, MEK, and small cell transformation. ADC, we can anti-HER3, HER2, MEK, or the TROP2. And then immunotherapy, I think we probably have to work on the tumor microenvironment, that's our strategy. And anti-VEGF in combination have demonstrated potential efficacy. And then they have been supported IMPA 150 subgroup analysis and Orient 031 data. And in the future, I think adenosine and anti-CD73 will hold high hope for the situation. That is actually all for today. But then one other strategy that I can share with you in the future is that when we go to buffet, what's the best way to have the best food? Thank you so much for your time. Thank you.